Hello and welcome to the Pinnacle 21 webinar on QC of SEND datasets. My name is Peggy Zorn and I am Senior Director of Product Management at Cetera, focusing primarily on the SEND Explorer dataset review and visualization tool. Just a little more information on me. I have a strong background in non-clinical data management, warehousing, and the SEND standard, including serving as the lead of the CDISC SEND controlled terminology team for more than 7 years. I continue to be involved in the CDISC SEND team as well as the PHUSE non-clinical working group. Prior to joining Cetera, I worked for 15 years at Pfizer, with 6 of those focused on non-clinical data management and warehousing. I also worked for INDS for several years, which is the company that originally developed the SEND Explorer dataset review and visualization tool. This is a list of topics we're going to cover in today's webinar. I'll be giving a brief overview of what the SEND standard is and how it developed, and we'll cover why having well-developed and documented QC processes for SEND datasets is so important, and what the major areas of QC are for SEND. We'll also be discussing best practices for QC of SEND datasets and the importance of using automated validation tools and SEND-specific review software to help make SEND dataset QC more efficient and effective. I'll wrap up the session discussing the key resources that are available to help those who are interested in more information, and then we'll take any questions that come in from attendees. SEND is the standard for exchange of non-clinical data. It is a CDISC standard based on the SDTM model and covers the study design and individual animal data for in-scope non-clinical study types. Like SDTM datasets, SEND datasets are produced in SAS transport or XPT format. The current version of the SEND standard in use at FDA are SEND 3.1, SEND 3.11, SEND IG DART version 1.1 for reproductive tox studies, and SEND IG AR version 1.0 for animal rule studies. The focus of this presentation will be primarily on SEND 3.1 and SEND 3.11. SEND datasets are required for single dose, repeat dose, carcinogenicity, cardiovascular and respiratory safety farm, embryo fetal development, and animal rule studies, with start dates after those published in the FDA Data Standards Catalog. SEND is required by both CDER and CBER. The requirement for CBER has just taken effect, and SEND datasets are now required for in-scope studies and data types for studies starting after March 15, 2023. SEND datasets for current studies are generally produced by the test facility, which could be a CRO or a sponsor, at the time of study report finalization for any studies that require SEND datasets for IND or NDA submissions going to the FDA. The majority of the studies requiring SEND datasets at this time for FDA submission are for IND submissions where timelines are tight. In many cases, work may begin on SEND dataset creation while the study report is still in draft stages. When this happens, it's important that as changes are made to the data in the study report that would affect the SEND datasets, those changes are also propagated to the datasets. This sometimes involves coordination across departments and even test sites if some of the data is coming from a location other than the test facility. Some sponsors also want to use SEND-specific review tools to review and visualize study data while the study is going on. In this case, SEND datasets that are not suitable for regulatory submission but can be used for review of the data by scientists working on the study may be produced at regular intervals until the study completes. There are also some sponsors that want SEND datasets for studies that are not going into FDA submissions, as they are using SEND as a data exchange and transmission format for feeding internal databases and data warehousing environments. SEND datasets may also be created retrospectively for studies that have already been completed and reports finalized, but it was not known that the SEND datasets would be needed. Sometimes in this case, SEND datasets are created manually from the study report tables when electronic source data is not available. In most cases, a combination of manual and automated processes are used to create SEND datasets. Data collected electronically in data collection systems can often be output directly to SEND format or fed directly into secondary systems that can produce SEND format. Most non-clinical data collection systems do not have all the fields necessary to fully output SEND trial domains, so often these domains have to be updated from the data output from the primary data collection system or created manually. For most studies, additional data collected outside of the primary data collection system used for standard in-life and post-life data also needs to be obtained and merged in with the trial domains and data output from the primary data collection system. There are vendor tools available to assist with merging, updating, and output of SEND dataset packages, but as you might imagine, this process can be complicated and sometimes produce unexpected results or errors. There are some studies where there is no electronically collected source data available, and the SEND datasets have to be created manually from the study report methods section in tables. There are a wide variety of extraction tools to assist with this type of creation process, and Excel is often used heavily to aggregate the data. The degree of automation possible in this scenario depends largely on the quality and format of the study report files. PDF files that are not searchable and cannot be OCR'd present significant issues. In addition to the datasets, the DEFINE.XML and NSDRG are also created for each SEND dataset package going to the FDA. Software tools to create the basic DEFINE file structure are used, and study-specific updates are then applied. NSDRG files are typically created using the PHUSE Microsoft Word template and manually updated to reflect the study, although there are some tools that can assist with auto-population of at least some of the NSDRG sections based on information in the SEND datasets for the study. We'll take a look at this a little later in this presentation. When considering the level of QC needed for SEND datasets, it's important to understand how the datasets were produced. For instance, if all datasets were created manually from a PDF study report, more QC would be needed than if the majority of the data was output from a validated GLP data collection system. In addition, SEND datasets are often produced by the CRO test facility that ran the study or a different third-party company that specializes in SEND dataset creation. The datasets are then passed to the sponsor for inclusion in the regulatory submission. In this case, it's important to the sponsor to have some understanding of how the datasets were created and what level of QC was done to determine what additional review might be needed. SEND datasets are used for a variety of purposes, and as the body of SEND datasets grows, so do the number of users and uses. First and foremost, SEND datasets are an FDA requirement, and so when provided in a regulatory submission, they are used by the FDA reviewers to review and analyze the study. SEND datasets and associated SEND review tools, which we'll be discussing in more detail later in this presentation, allow reviewers to sort, filter, summarize, and visualize the study data quickly and easily. In addition to the FDA reviews, scientists working on the non-clinical studies, including study directors, pathologists, TK scientists, and more, are starting to use the SEND dataset format earlier in the study process to review and interpret the data. SEND datasets have also become the leading format used for population of non-clinical study data warehouses that support and facilitate cross-study analysis. Again, SEND review tools have been optimized to read, display, and visualize this format and are really revolutionizing non-clinical study data review and interpretation. The level of QC needed on SEND datasets may also vary based on the intended use of the datasets. For datasets going to the FDA for submission, full conformance with the SEND data standard and the FDA Technical Conformance Guide would be expected, while SEND datasets meant only for sponsor use in a data warehouse environment may have different requirements. So why are QC processes so important for SEND datasets? In this age of automated systems and software, isn't all of this just output from an easy button in some system with no additional worry or work needed? Well, as you heard in the previous section, SEND datasets are created using a combination of automated and manual processes in most cases, and integration of data from several sources is usually required. Trial domains are often created manually, and in some cases, the SEND dataset creation process may be done entirely from the PDF study report file. During the SEND dataset creation process, the study report tables and dataset output are often compared to ensure alignment, something that we'll be talking about further in this session. During this process, errors are often discovered and updates are then need to be made. In addition, the DEFINE.XML and NSDRG files are also created using a combination of automated and manual processes and need to be synchronized if dataset changes are made after their initial creation. Often CROs or other third-party vendors are responsible for SEND dataset creation and at least partial initial QC of the data they're responsible for, and datasets pass to the sponsor for inclusion in the regulatory submission. All of this adds up to a process where changes to the datasets could be happening throughout the creation process, and at least some of the source data for the datasets may have been created manually. Because of these factors, a robust quality control or QC process for SEND datasets is essential to ensure high-quality datasets for regulatory and scientific use. In this section, we're going to discuss other major reasons why SEND dataset QC is so important. The FDA Technical Conformance Guide emphasizes the need for strong and systematic dataset QC practices. In two different sections of the FDA Technical Conformance Guide, the need for QC processes are stressed. Section 8.2.2 indicates that sponsors should evaluate their study data prior to submission against conformance rules published by CDISC, the ECT Technical Rejection Criteria, and the FDA Business and Validator Rules. FDA expects that any issues found be corrected or explained in detail in the reviewer's guide so that consistency and traceability between the study report and data set is ensured. In Section 8.3.1 of the Technical Conformance Guide, FDA further emphasizes the importance of the traceability between the study report summary and individual data tables and the data sets, and recommends that data collection systems have a well-defined relationship to the SEND standard. In addition to the statements in the Technical Conformance Guide, FDA routinely does presentations to the non-clinical community regarding quality issues with SEND datasets, including information on those issues that most interfere with submission reviews. This has been done in the past as part of the Small Business and Industry Assistance, or SBIA, webinar series, as well as through presentations done as part of the CDISC SEND full team meetings, through Fit for Use pilot feedback, and through several presentations done to try and publicize and educate SEND dataset providers on the FDA technical rejection criteria that was implemented over a year ago. The PHUSE non-clinical working group took on a project a couple of years ago to investigate what type of QC practices companies are using for their SEND datasets, and produced a white paper and poster that was presented at the last PHUSE Computational Science Symposium meeting. This also emphasizes how central the issue of SEND dataset QC and quality is. This team outlined results from a survey they had done of non-clinical group members in the PHUSE network on QC practices they felt were important and actively perform. There were 51 responses received, most from the US, but feedback was also received from the CDISC Japan users group, who reviewed and commented on the survey results to provide a more global perspective. The PHUSE non-clinical working group subteam identified 6 major areas that characterize high-quality datasets that include complies with regulatory requirements and guidances, including the TCG, the TRC, the ECT submission specifications, and more, conforms to the SEND IG, correctly represents the study, which means aligns with the study report, consistent across datasets. This means for studies for the same compound or using the same methods, values are consistent where they should be. Understandable, which means the NSDRG contains study-specific explanations for any study or data anomalies or areas where the report and the datasets are inconsistent. And fit for use, which means reviewers and scientists can use the datasets for review, analysis, and interpretation. These areas are important to keep in mind when developing SEND dataset QC approaches and processes. Now it's clear that SEND dataset QC is a major issue for dataset creators and a priority for SEND consumers, including FDA. Let's talk about some of the major areas involved in QC of a SEND dataset package. First and foremost, SEND datasets are required to comply with the technical specifications included in the SEND implementation guide. Current versions of the SEND IG being accepted by FDA are SEND 3.1 and SEND 3.1.1 for general toxicology and cardiovascular and respiratory safety pharmacology studies, SEND IG DART 1.1 covering embryo-fetal development studies, and SEND AR 1.0 covering animal rule studies. SEND IG 3.1 provides a list of items that must be met to achieve minimal conformance with the SEND standard, and these items apply to all versions of the SEND standard. These items focus on dataset completeness, structure, conformance, and format. To be complete, SEND datasets must include all collected and relevant derived data in the standard domain supported by SEND. The dataset should include all required and expected variables, and all required variables must be populated. In addition, each domain record should include appropriate identifier, timing, and topic variables. The structure of SEND dataset should follow the metadata structure defined for each domain and follow the models included in the SEND IG. The SEND datasets must conform to the specifications included in the SEND IG, including use of the SEND IG specified standard domain names and variable names and labels, application of specified SEND control terminology, and conformance to the business rules described in the domain's assumptions and CDISC notes sections for each domain. The format of SEND datasets must align with control terminology, and all datasets must be output to SAS Transport or XBT version 5 format. In addition to the SEND implementation guide specification, SEND datasets should also conform to FDA guidance published in the FDA Study Data Technical Conformance Guide. This document is updated at least a couple times a year, usually March and October, and gives detailed guidance on specific areas that are recommended to facilitate regulatory review. In addition to dataset-specific recommendations, the FDA Study Data Technical Conformance Guide also includes detailed information regarding requirements for placing datasets correctly within an electronic common technical document or ECTD submission, and information regarding requirements included in the FDA Technical Rejection Criteria guidance, which when met, allows submissions containing standardized studies data to be automatically processed correctly through the FDA Electronic Submissions Gateway. The FDA Data Standards Catalog is a spreadsheet that contains all of the past and current electronic data standards used at FDA, including details on which versions are currently accepted at which centers, dates when requirements begin and end, details on which submission types they apply to, controlled terminologies that apply, and more. This file is updated throughout the year as needed. For instance, 3 versions were issued in 2022 and 1 so far in 2023, so watch out for updated versions of both the Study Data Technical Conformance Guide and the Data Standards Catalog for the latest FDA information on standardized study data. Both FDA and CDISC have published rule sets that cover SEND datasets largely based on information in the SEND IGs, the Study Data Technical Conformance Guide, and other associated FDA guidance documents related to electronic submissions. These rules form the basis of what checks are included in automated validator tools available for use with SEND datasets. The FDA Business Rules version 1.5 are divided into rules that apply to both clinical and non-clinical, and those that apply only to clinical or non-clinical. Many of those related only to non-clinical deal with detailed rules around alignment of SEND datasets with the tumor.xbt file, which is also required for carcinogenicity studies. The primary focus of the FDA business rules is to provide rules for major areas that support successful regulatory review. The FDA validator rules are much more detailed and comprehensive in nature and include support for SEND 3.0, 3.1, 3.1.1, SEND IG DART version 1.1, and SEND IG AR 1.0, as well as rules associated with the Study Data Technical Conformance Guide, the FDA business rules, ECTD and Technical Rejection Criteria rules, and also CDISC SEND Conformance Rules version 4.0. The FDA Technical Rejection Criteria discussed previously contains rules related to successful processing of submissions containing standardized study data through the FDA Electronic Submissions Gateway. The CDISC SEND team has a rules subteam that has also published conformance rules based on the SEND implementation guides it covers, currently for version 3.0, 3.1, 3.1.1 for general toxicology, carcinogenicity, and cardiovascular and respiratory safety pharmacology studies, and DART version 1.1 for embryo fetal development reproductive toxicology studies. While the FDA validator rules appear to cover all of the other items on this list, it's a good idea to make sure that any automated validator tool you use has coverage for all items on this list. FDA has said in the past that they would like to get out of the rules business for standardized study data, and that detailed conformance rules for datasets should be published by the Associated Data Standards Organization at the time the implementation guide is published. CDISC has moved in this direction, so in the future we may see less leadership from FDA in this area and more from CDISC. But for now, be aware of all of these sources for conformance rules that are relevant for SEND datasets and submissions containing SEND data. The final study report serves as a comprehensive and definitive source of information for non-clinical studies and includes both summary and individual data listings for all data collected and analyzed in the study. SEND datasets associated with non-clinical studies should align with the information and data included in the study report wherever possible. The importance of this alignment is discussed in the FDA Study Data Technical Conformance Guide and was the guiding premise for the work done by the PHUSE non-clinical working group that looked closely at SEND dataset QC practices. There are several important areas of alignment between the study report and SEND datasets that should be included in any SEND dataset QC process. First, the study design outlined in the study report or protocol should align with the trial design domains in SEND. The trial summary or the TS domain should be reviewed carefully, as many values included in this domain are used as grouping and metadata values for the results. Most importantly, all animals included in the study should be represented and associated with the correct treatment group and sex. All data types included in the study report in scope for SEND should have associated data sets. The study report individual data table should align with the corresponding SEND dataset content, including any exclusions, comments, reason for results not collected, etc. Unscheduled or unplanned events should be represented correctly. This is a big one, as often unscheduled events in a study cause issues with data collection and reporting that can carry over to inconsistencies in the SEND datasets. Most importantly, SEND datasets should be able to be used by FDA reviewers to replicate data summarizations included in the study report. Any important differences between the study report and the SEND dataset should be explained in the NSDRG. In addition to QC of the SEND datasets, for SEND datasets included in FDA submissions, QC of the required DEFINE.XML and NSDRG also needs to be accounted for. QC of the DEFINE file should be based on the DEFINE standard. 2.0 or 2.1 are in current use, with all studies starting after March 15th of 2023 required to use version 2.1. Some automated validator tools include DEFINE file support, and this is an important factor in choosing a validator tool to use with your SEND dataset package, because the underlying XML structure and dataset metadata has specific requirements that are best verified using automated tools. In addition, DEFINE.XML files should contain information specified in the SEND IG and TCG, such as an indication if the value in EX dose variable in the EX domain represents planned or actual dose levels, the definitions for RFS to DTC in the DM domain, and the experimental start and experimental end parameters in the TS domain, and the full severity scale used for microscopic findings. DEFINE files should also be study-specific, so things like domain structures and keys, comments, and computational methods should reflect the study and not just templated text. The NSDRG is an important piece of a SEND dataset package for submission. The NSDRG should follow the latest PHUSE template, should be study-specific, and should clearly explain any important nuances in the study that affect the datasets and any significant differences between the study report and datasets. The NSDRG should be checked for errors, typos, and the final NSDRG.pdf file should comply with the FDA PDF specification guidance document. QC of SEND datasets and associated files like the DEFINE.XML and NSDRG are a critical part of producing a high-quality SEND dataset package that is able to be used effectively by FDA reviewers and non-clinical scientists. This last section of the webinar focuses specifically on best practices for process development and use of software and automated tools to help make QC of SEND dataset packages easier, more consistent, and produce the best possible product for SEND consumers. In developing an overall QC process for SEND datasets, there are several key areas to focus on. Because SEND dataset creation often involves integration of files from different source systems and some manual data creation, and because SEND dataset creation can often start while a study report is still in draft stages, it's important to ensure that QC is occurring at various milestones throughout the SEND dataset creation process. Keeping detailed records of issues identified, changes made, and copies of validation reports from defined review cycles is recommended so that final review of the dataset package can flow smoothly. As with other quality control processes, it is important that another set of eyes be involved. The person doing the QC of the dataset should be different than the person creating the datasets. While self-QC is an important part of the overall process, having a different person or persons performing the final QC check will result in a much more robust and valuable process. We've discussed how important it is that the study report and the SEND datasets align, so it is critical that the study report, including all summary and individual data tables, be available for reference during the QC process. Use of an automated validator tool is a must. It should be run and results evaluated after every major dataset or DEFINE file update cycle. Validator tools should provide comprehensive rule set coverage and output issues in a logical, understandable way that links to the associated rule. The most useful dataset validation tools provide enhanced support for issue tracking and resolution, issue libraries with standard actions and explanations, and even automated production of DEFINE.XML and NSDRG files. Ensure that the DEFINE file is created against the final version of the datasets whenever possible. DEFINE file creation tools gather required metadata from the datasets themselves, and if updates are made to the datasets, that metadata will change and have to either be updated or recreated. We've talked about the value of using automated tools whenever possible, and this is definitely true for both the DEFINE.XML and NSDRG files. For the NSDRG, there are several areas and sections that can be populated directly from the datasets, define file, or validator output, and doing this automatically can significantly reduce effort and errors. To ensure consistency in the QC process between QC reviews in and across studies, it's important to have any necessary manual QC steps documented in a shared format for use by all staff involved in QC of SEND datasets. And finally, it can be challenging to review, QC, or even open SEND datasets if you are not using a review tool specifically optimized for the SEND format. Tools such as the SAS Universal Viewer and Excel can help, but SEND-specific review tools often contain robust sorting, filtering, and query options and displays specifically designed to facilitate QC, such as tabular summary tables with flexible grouping and display options. SEND-specific review tools also can provide easy linking to related records in the comments, supplemental qualifier, and related record domains so that these domains can be easily verified. Because automated validation checks are such a critical piece of SEND dataset and define file QC, I wanted to spend just a little more time focusing specifically on things to look for in SEND validation tools. First, ensure that the tool has comprehensive coverage of all the major rule sets from FDA and CDISC, and that it is up to date with the latest versions. For instance, version 1.6 of the FDA validation rule set was updated in December of 2022. An automated validator tool should test against all relevant rule sets and also flag inconsistencies across domains. For example, animals not included in the demographics domain should not have results in any findings domains. The validator should be able to report any issues related to application of SEND-controlled terminology using the version referenced by the TS domain for the study. The list of validation issues output by the validator tool should be comprehensive, logically organized, and provide clear reference to applicable rules and standards. As validation can and should occur throughout the dataset creation process, a tool that can aggregate results across reports and clearly show new issues is a huge time saver. Many of the same validation issues occur across domains and studies, some of which will not be resolved and must be explained in the NSDRG. Validation tools that can store issue libraries that include help text for fixing issues and standard wording for explanations on issues that will not be addressed can significantly streamline issue resolution and population of the validation issues section of the NSDRG. It is extremely important that the validator tool cover both the datasets and the DEFINE.XML file, as manual QC of the DEFINE file underlying structure is very difficult. Some validator tools can also automatically create the DEFINE file in NSDRG from the dataset package, saving significant time and reducing errors. This screenshot shows a validator report for a SEND dataset that includes issue tracking across multiple validation runs. As you can see, it notes number of new, fixed, updated, and unchanged issues since the last run, and provides a logical, easy-to-use dashboard of the validation status for the study. It is important that validation reports be logically organized and easy to understand. This validation report is organized by SEND domain, includes the rule ID, the current status, and a system-assigned impact value that helps with prioritization of work. These screenshots show a validation tool report that allows issues to have explanations added from an internally maintained, customizable library, which promotes consistency and streamlines issue resolution and NSDRG production, as all validation issues that will not be fixed must be reported with explanations in the NSDRG. A SEND dataset reviewer tool is another critical piece of the SEND dataset QC process. Because final SEND datasets are output in XPT format for submission to FDA, it can be difficult to open and review the data using standard tools like Excel. There are some dataset creation tools that can output other formats such as CSV, XLSX, and XML in addition to the XPT format, but even using Excel for QC of datasets can be extremely time-consuming, tedious, and ineffective. Review tools designed specifically for SEND datasets have really important features that make QC tasks much easier and more effective. SEND review tools should have advanced filtering, sorting, and column management features, as SEND datasets can contain many columns and rows. Being able to filter and sort across an entire dataset to look for anomalies, missing data, and data inconsistencies is key to any dataset QC process. Some tools also have the ability to merge in metadata about an animal with the result, such as sex, dose level received, group number, disposition type and day, and more, so that results can be easily grouped and summarized in ways that align with the study report tables and displays. In addition, tools that allow easy linking between related domains such as finding comments, supplemental qualifiers, and related records make QC of these related domains much easier. Because comparisons between the study report and the datasets are a key component of dataset QC, review tools that have flexible reports and data presentations that allow for easy comparison to study report tables for checking means, ends, and standard deviations are critical. Some advanced SEND review tools also include features such as the ability to generate CTD tables and access to data visualization displays that allow scientists to interactively use and interrogate study data to find trends and patterns within and across data types. This is a screenshot that shows a SEND dataset and a review tool that provides a flexible data grid that includes metadata such as group number, sex, dose level, and disposition type integrated with the findings data. In this data grid, all the columns can be easily sorted and filtered, and additional columns added and removed from the display. Notice the subject number is hyperlinked, and when clicked, takes the user to a specialized display that includes all data and details for the selected subject. Selecting the pivot or summary buttons allows users to summarize the data using a variety of options for easy comparison to study report, summary, or individual tables. This screenshot shows a SEND viewer that has direct record linking between related domains. In this case, the user can click on the hyperlink in the comments column in the microscopic findings domain to see any comments associated with the result. Having this linking in place directly in the parent domain makes QC of domains such as the microscopic findings domain much easier, as many results have linked comments, result modifiers, and linkages to the macroscopic findings domain. If they are not able to be viewed together in a viewer, then the related records must be traced manually to confirm correct linkages between the domains. The next couple of screenshots show a study schedule view that can be filtered by data type or domain to see counts per group. The view can then be drilled down all the way to the animal level to see individual values. This is an excellent way to look for missing or duplicate data and other data anomalies. This screenshot shows dosing records, where it's easy to see that dosing may have stopped for the high dose group earlier in the study, and then compare it to the study report or protocol language to confirm the data is correct. This next screenshot shows drill down to body weight data for individual animals and time points for easy comparison against study report tables. This is a clinical pathology tabular display generated inside the SEND review tool that can be flexibly sorted or grouped to align with the study report summary tables for quick and easy confirmation of group summary values, means, standard deviation, ends, etc. The format shown is sorted by test, then time point, but that can be changed to time point, then test, and filtered by category, and a wide range of other SEND variables to make the SEND review tool report align with the format used in the study report tables. These next two screenshots show a SEND review tool that is able to generate two of the key tables that are included in the NSDRG, the Section 2.2 Trial Design Domain Overview Table and the Section 4.1 Dataset Summary Table. Both of these tables are meant to reflect actual structures and files included in the datasets, so it is ideal that the tables be generated from the datasets automatically by a system rather than hand-entered in the PHUSE Microsoft Word template. This is a Section 2.2 Trial Design Domain Overview Table. And this is a Section 4.1 Table of Domains actually included in the SEND dataset package. Using dataset validator and review tools that include functionality to automatically generate large portions of the DEFINE.XML and NSDRG are excellent ways to reduce the QC burden and ensure that these accompanying files align exactly with the SEND datasets. Even with the use of automated validation tools, there are still checks that need to be done at least partially manually. The QC reviewer will need to review the output produced by the validator tool and ensure that any issues that can be resolved have been resolved, and any issues that can't be resolved are explained appropriately in the NSDRG for both the datasets and the DEFINE.XML file. The trial domain structure and metadata domains, the DM, the DS, the SE, form the backbone of the data sets and have a huge impact on how this data can be summarized and analyzed. A thorough review of the trial and metadata domains is very important, as if the dose levels are inaccurate or animals are assigned to the wrong sex or group or are missing, the ability to use the data for any kind of review or analysis will be significantly impacted. To ensure alignment between the study report and the data sets, QC review usually focuses on comparing the study report method section and summary and individual tables to the datasets. This can take the form of spot checking, but generally a more standardized approach, for example, review all data for one animal per set per sex, or review a certain percentage of data in each domain, for example, 5 or 10%, or comparison of summary tables generated from the datasets to the study report summary tables is done. For very large studies, this summary review could focus on a random sample of time points or tests or tissues. For these types of reviews, using a SEND review tool that can flexibly generate summary views of the data for comparison to report tables is very helpful. Checklists to guide manual review processes are excellent tools to ensure important QC steps are not missed and to promote consistency between different reviewers. When QCing a SEND dataset package, DEFINE.XML and NSDRG files should also be a part of the review process. As mentioned in the last slide, if these files are largely automatically generated by the validator or SEND viewer tool, then the QC process will be easier and can focus on those portions of the files that are updated manually. For the DEFINE.XML file, it's important to note that the DEFINE file should be created against the final version of the datasets. If updates to the datasets have been made since the DEFINE file was generated, updates will need to be made to address changes, or if the dataset changes were extensive, the DEFINE file may need to be recreated. QC of the DEFINE file should always first include running an automated validator tool and reviewing and resolving any issues found. The SEND implementation guide has recommendations for several key areas in the DEFINE file that should be updated based on the study, including definitions for RFST-DTC in the DM domain, the experimental start and end dates in the TS domain, the EX dose variable in the exposure domain, comments for all null expected variables, and the full severity scale for microscopic findings, and more. These areas may have been manually updated in the DEFINE file and will likely be study specific, so should be a focus during QC. In addition, it's a good idea to spot check hyperlinks to ensure they are relative and that the link to the NSDRG is present and functioning. QC of the NSDRG should ensure that the file follows the PHUSE NSDRG template and all sections are populated correctly. Check the table of contents has been updated to reflect the content of the file and that no errors are present. As with the DEFINE file, the content of the NSDRG should be study-specific, and any template text replaced with study-specific wording or removed if that text is not relevant. The final NSDRG PDF file should follow the specifications included in the FDA PDF specification guidance documents, including bookmarking, hyperlinking, and font and other formatting requirements. Some datasets have become an integral part of the review, analysis, and interpretation of non-clinical toxicology studies, and as study types and users are added, this trend will only continue to grow. The SEND dataset creation process is still challenging and often requires data integration and some element of manual effort, making quality control a critical step to ensuring that the SEND dataset package for a study is fit for use. The level of QC needed on a SEND dataset can depend on the dataset creation methods used and on your role in the process. For instance, if you are a dataset creator and the datasets were created manually from the PDF of a study report, the QC process needs to be very comprehensive. On the other hand, if you're a sponsor company that is receiving a SEND dataset package from a CRO where QC has already taken place, it's important to establish confidence in the CRO's QC process for the SEND dataset package and to tailor additional QC steps on the sponsor side to focus on areas of most risk. At a minimum, an automated validator tool should be run on the datasets in DEFINE file, the datasets opened and spot checked in a SEND review tool, and the NSDRG checked for accuracy. As mentioned, there are automated tools that can make the quality control process more effective and efficient. Use of an advanced validator software tool and a comprehensive SEND review tool are key to developing a robust and sustainable quality control system. This is a list of helpful SEND dataset, DEFINE.XML, and reviewer guide resources to help in QC of SEND datasets. I've included links to all the major items mentioned in this presentation, including the PHUSE, FDA, and CDISC resources. That concludes the content portion of the webinar. Now we'll take a look at some of the questions that have come in from attendees and try to answer as many of those as we can. So the first question we have is, what version of FDA rules are implemented in the Pinnacle 21 validator? The latest rules used at FDA, including version 1.6 of the FDA validation rules that was updated in December of 2022, are included in the Pinnacle 21 Enterprise and Community Validator products. Next question, do you have any suggestions on expected documentation for QC processes? Any ideas on the format, content, or level of details that should be included? Well, the documentation for QC processes is important as it can provide a record of what was reviewed, how it was reviewed, and the updates that were made during the review cycles. At a minimum, versions of issue reports from automated validator tools are really helpful to have around, and validator tools that allow easy tracking and comparison of issues between reports and the resolution status provide really good documentation of the dataset validation process that was done for a dataset package. As I mentioned in the webinar, checklists are another good way to document the overall QC process, including any manual checks that need to be done, and they really help promote consistency across reviewers and projects. All right, next question is, what is the difference between SEND and SDTM? All right, SDTM, or the Study Data Tabulation Model, defines a standard structure for human clinical trial study data tabulations and for non-clinical study data tabulations that are to be submitted as part of a product application to a regulatory authority, such as the FDA. The Submission Data Standards team of the CDISC, or the Clinical Data Interchange Standards Consortium, defines SDTM. SEND is based on the SDTM model and provides support for non-clinical studies. So basically, SDTM is the base standard, the base data standard and model, and SEND is an offshoot of SDTM designed to handle non-clinical study data. This next question is related to the validation for tumor.xbt files. Does Pinnacle 21 have any plans to offer validation for the tumor.xbt together with the SEND package or as a standalone? So right now there is a FHUSE workgroup forming to develop the first set of conformance rules for the tumor.xbt specification. These rules will be incorporated into the Pinnacle 21 validator shortly following publication. Next question, what tool does FDA use for validation and viewing of SEND data sets? The FDA currently uses Pinnacle 21 Enterprise for validation of SEND data sets and SEND Explorer Warehouse for viewing, analysis, and visualization of SEND data. All right, so the last question we have for today is SEND Explorer free? How can I download it? SEND Explorer is a licensed product for viewing, data analysis, and visualization of SEND datasets offered by Satera. You can find more information on SEND Explorer by visiting the Satera website at www.satera.com. Thank you for your attendance at this webinar. Just a reminder, we will post the recording and slides in the next couple of days, and you will receive an email notifying you when this has occurred. If you posed a question in the chat and we did not have time to answer it, we will be in touch to follow up with a response. If several people pose the same question, the answer will be included in a post-webinar blog article. My contact information is below, and please feel free to reach out to me if you have any further questions or want to learn more about the tools available to help with QC of SEND datasets.